Good morning, everybody. So welcome to the virtual regenerative medicine workshop, part of the webinar series, Next Generation Organ-on-Chip and Organoids. Regenerative medicine includes gene therapies, cell therapies, and tissue-engineered products intended to augment, replace, or regenerate organs, tissues, cells, genes, and metabolic processes in the body. Welcome to the second workshop of the Next Generation Organ-on-Chip and Organoids webinar series focused on regenerative medicine and organized jointly between CSEM and AMIRES. I'm with Jill Beda, who is a team leader at CSEM Tools for Science, Life Sciences, developing innovative solutions for organ-on-chip, tissue engineering, point of care, diagnostics, and the lab of the future. Jill, can you please tell us why the regenerative medicine topic is hosted by the Next Generation Organ-on-Chip and Organoids workshop?
Sure, Mariana. Thank you. So the main goal of this initiative is to bring together emerging technology and big players from the pharma and biotech industry under the same roof or behind the same screen. Emerging technologies feed the development of innovative solutions for safe and effective regenerative medicine. At the interface and lab and clinic, you will learn more about the OrganTrans Consortium gathering technology providers, medical centers and research institutions, patient organizations and therapeutic developers. Strong community and communication are needed to overcome barriers associated with the adoption of new methods in a regulated environment. Mariana, who is Program Project Manager in Health and Biotech at AMIRES, will tell you more about the format of the next 2 hours.
Yes, yeah, thank you. So we have 3 sessions. The first of them is with the technological solutions in regenerative medicine. We have speakers from CSEM, Google Myers, Regeneration, and IBD. Then we have a session about OrganTrans and AMIRES as a funding enabling regenerative medicine topics. And then the third session is about the therapy solutions in regenerative medicine with ContiPro, SpinPlan, Bionova, Cutis, and Foundation, Don Carlo Gnocchi Foundation. For timing, we opted for multiple but short talks. Therefore, we ask each speaker to have maximum of 7 minutes. Then we will be informing you about these 7 minutes, and you will have 30 seconds to conclude before we remove your slides. Please, to all the attendees, do not hesitate to post your question in the chat, and that will be visible to all speakers. We will address the questions and answers at the end of each session, depending on the remaining time. If during the event we cannot address all the questions, we will try to follow them up individually. So therefore, we encourage that you write your name.
So before entering to the heart of the matter, I would like to highlight 3 keywords: closed system, standardization, and regulation. Today, we will hear about the increasing importance of these 3 keywords from technology providers to clinical applications and adopters. Finally, we are extremely happy to see the equal distribution of participants between university, research technology organization and company, while hoping to see an increase of the clinical participants in the next years. Now, it's my pleasure to introduce you the first speakers, my colleague from the Tools for Life Science, Vincent Révol from CSEM.
Thank you, and let me share the screen that you should be able to see it now. Thank you for your short confirmation.
Yes, we can see it.
Okay, thank you. So good morning. It's my pleasure to introduce in a short speech our activities around Tools for Life Sciences. Next generation sequencing, wearable devices, ELs, organ-on-chips, and many other examples illustrate the increasing importance of advanced technologies for life science applications and healthcare. If we look at the field of regenerative medicine, We have, on the one hand, high potential for new therapies, going over gene, cell therapies, organ replacement, and we'll see many of example of this therapeutic potential in the next presentation. Some of these therapies are already in clinical application, but many of them are still in the pipeline, and they are facing some challenges to, of course, reach the patient and the clinics. Some of the challenges are related to standardization, automation, sterile system that basically make you also less dependent on the operators and be able to produce at the right cost for making sure that the patients can pay also, or the health system can pay for such therapies. There are also several challenges related to quality control and finally making technologies that are compliant with general manufacturing practices, GMPs. And finally, a specific challenge related to scale out and sometimes scale up to be able to also be able to produce at decentralized location. For some of these challenges, new technologies are required, and this is where CSEM comes in. As a Swiss technology provider, our mission is to develop new technologies and to facilitate the transfer of these technologies to industrial applications. So in a sense, we are building a bridge between academic science and industrial application. In the field of life sciences, we have a large heritage in micro and digital technologies, and we capitalize on this heritage together, of course, with knowledge on biology to develop new license application. We have three main pillars: cell technologies, how to grow cells, handle cells; biomonitoring, to measure activities of cells; and of course lab automation, to automate the process, handle the cell automatically and make sense of the data. All of these competences are found of interest, of course, when you talk now specifically of bioprocesses. We have identified several technology blocks that we find are very similar or can be found in many of the different processes. You usually start with some cells or biopsies. You want to standardize the production. You may want to isolate or sort these cells according to quality criteria. Finally, you want to assemble them or formulate these cells into the final product. All the bioprocesses in regenerative medicine are often taking days or weeks, so you need a stringent maturation and biomonitoring process technologies to monitor this. And finally, you have some endpoint analysis. Interestingly, these building blocks can be found both, of course, for drug discovery application as well as for regenerative medicines. In the past years, we have proven the ability to really deliver from the technology products, and you see here 3 selected examples in the area of some standardized production with Sun Bioscience, in the area of sorting for especially zebrafish in this case with Ulmos Institute, and finally with Novartis in the area of maturation of the tissue and biomonitoring. I'd like to dive closely on 2 specific examples. The first one is a technology platform to enable the sorting of cell organoids or organisms. So we really go from individual cells, 10 micrometers, up to large organisms that can be zebrafish or other type of models that are used in drug discovery. And to enable this, we have 3 main aspects. One aspect is related to the use of MEMS and microfluidic technologies to handle this cell in an appropriate way. So being able to also maintain the viability, not changing any of the cell expression and so on. The second aspect is related to the use of deep learning, and this is a specificity of CSEM to combine these 2 technologies together to classify the cell according to your criteria. And finally, the overall automation to run this process without any operator. One example of this technology platform is a collaboration with the company Cellular Highways. They have a vortex cell sorting principle that we together developed into a microfluidic chip. You see this picture, and impressively, this microfluidic chip can sort up to 40,000 cells per second. You can imagine how fast that is. And this is really enabled by the combination of, again, micro system technology and the knowledge about liquid handling. Another example, now going toward larger entities, is for organoids. You see here the example of organoids that are grown into the plate from Kugelmeyers. They are roughly 80-100 micrometer in size. On the left video, you see the classification of these organoids according to green and red, so basically good or bad organoids. And on the right side, you see videos of a technology able to also individually pick single organoids into such a microarrays and extract them if they don't have the right quality standards. Again, illustrating these key technologies can be used in several bioprocesses. The second example or highlight is on a collaboration together with Novartis and several other companies to create automated contractility assays for micromuscle. And this is used for studying skeletal disease.
Vincent, you have 7 minutes already.
Okay. So basically, this illustrates the combination of the customized labware and physical environment and the automation of that. And finally, now, these technologies are nice, but when we go towards regenerative medicine, we have to consider GMP compliance. There are 4 elements that are important when you go into these directions. The use of a closed system and to automate the process in a closed system. The use of aseptic connectors and disconnectors. To be able to continuously and remotely monitor your process, and finally to automate the process and ensure data traceability. So with all that, thank you for your attention, and if you want more information, you can look at the license brochure of CSEM.
Thank you very much, Vincent, for this presentation. So according to the agenda, we jumped directly to Patrick Kugelmeyer from Kugelmeyers.
Thank you very much for giving me the opportunity to having the talk with you together in this regenerative medicine workshop. I just would love to have the controls so that I can share my screen. I need to see how I can do that.
On the top right, close to the leave, you don't press the leave button, but you can arrow, and then you can share your screen.
Thank you so much. That's great. Can you see already?
Perfect.
Fantastic. And the sound quality is good as well.
Excellent.
Okay, perfect. Thank you so much for giving me the opportunity, dear participants. This is not an ego problem to be the company named Kugelmeyers. We just were thinking, what are we doing? We are making spheroids, and Kugel in German means spheroid and Meyer means manager. So we are some kind of spheroid managers. And then we thought, okay, we need to take the name for the company name. So we are coming from, sorry, there was just a jump. We are coming from applied clinical cell transplantation. What you are seeing here is the pancreas. So this is a huge gland in the human body producing mostly digestive fluids. However, in the miracle is a little portion of these cells, only 2% of the entire mass are cells that are producing actually insulin. So if you are diabetic, especially type 1, your insulin cells that secrete insulin do not work anymore, and so you have diabetes. Let's have a look how these cells look like. These cells are organized as spheroids, little mini organs, let's say, or organoids, and they are clustered with about 200 to 2,000 cells, a mini organ, and hundreds of thousands of them are located in the human pancreas. So people were thinking, well, if it's only that few cell mass, we could just extract these from the pancreas and then just inject them into the liver of a patient. That's super elegant, and it's actually being done since more than 20 years. It's called eyelid transplantation. You have to imagine this. The people, they are awake, the patients are awake, they're just getting local anesthesia, you inject the cells they need, and the next day they go home. In an ideal world, they will be cured. But the problem is, when we have a look, well, how do these mini organs in the new host now look like? You see they are disconnected from the surrounding tissue. What does that mean? Previously, these organoids had massive vascular network. And of course, if we isolate these hundreds of thousands, then you cut actually this network. So there's no blood flow anymore. And there comes a fundamental principle of how to work with organoids and spheroids in general, because there is now coming the laws of diffusion. When you want to have oxygen to reach every cell, nature invented a vascular system. You can check this with your finger. Take a knife and check if there is blood. And you will see that there's everywhere a vascular system needed. And if not, you will have a diffusion limitation and the large spheroids will die because of hypoxia. So we would show that only small islets survive in that clinical transplantation, and that they are performing up to 3 times better than if you have, when you have by chance a donor who has more small islets. So that led us to the thought, well, if we have just a physical problem of diffusion limitation, and that's the reason why this transplantation doesn't work very well, we need to change something. We have the raw material of these islets, and we know that small ones perform better. So let's do something. We will dissociate them into single cells and re-aggregate them to be super standardized, all the same size and optimized for clinical use. So we developed our platform, the Spherical Plate 5D. And this platform allows to create absolutely regular spheroids with uniformity, functionality, and scalability. So why do I say that? There is this principle that you need to assure that all your further work, being that diagnostic or therapeutic and also just research, is doing well. So the spheroids need to be uniform because of diffusion distance and of regulatory pathways. They need to be functional, of course. If the tissue you're transplanting doesn't work, and especially for later in regenerative medicine, if this tissue doesn't form because it can't develop the correct way, it will not work and can even be dangerous. And you need to be scalable because the lab, of course, is small scale and you need to be large scale for patients. So we need to bridge that gap. So we will be doing a clinical trial, and let's have a look how this plate is looking like. So you see here our patent, there's a square opening on the top and rounded tips on the bottom. So we assure that every cell finds its partner, because there is no way where they can lay around. And we have a special coating, and like that, with the rounded tip, we assure that there's optimal forming of the spheroids. So we have a laboratory product created to bridge the gap between clinic and lab, and you have with one pipette tub, for example, 750 microwells, 750 spheroids formed, and with the medical plate that is not yet on the market, but you can, if you need it, you can just give us a call, and you can then create a lot of spheroids in absolute regularity. Just a little principle, we built in some laws of developmental biology, like morphogen gradients, that need to be assured that the development is happening correct, which is important from a geometrical point of view, because fate decisions are done by concentrations of signaling molecules that create your fate of the cell. So we believe currently that there is no alternative on the market to have this proper signaling guaranteed while being applicable for medicine. So I see I have a little bit short of time, so I will speed up. You see here that this is the largest megatrend in medicine. If you enter spheroids in PubMeds, there's nothing steeper and exponential than that. This is fascinating because this leads to a totally new world. We have had 3 silos of science, diagnostics, and clinics until now, but now there's happening a convergence towards a new field in life science, actually, that is a platform ecosystem with spheroids as common ground. And like Vincent beautifully showed before, you need a platform for that, and you can see that in your daily life. If you take your phone, this has now a convergence of all these materials that you previously had. We also want to contribute to that ecosystem, that anybody who needs to have a high quality.
I'm sorry, Patrick, you are 7 minutes.
Yes, exactly. 30 seconds left. So I will be ready in a second. So yes, there is an ecosystem of players that you can have a one-stop shop for any application, and it's building like that right now. And with that, sorry, with that, we will be having a totally new form of medicine where you add health instead of repairing, because you can just replace the tissue that is not the right way. And the entire world is working on that. And we here as a group want to assist in making these solutions possible. And I thank you very much for your attention.
Thank you, Patrick. You also planned the end of the talk with the ring, so it is perfect on time. Thanks. So
Thank you.
We directly jump to Mauro Petretta from Regeneration.
Hello. Thank you. So I will try to ask for control.
Okay, let's stop. Yes.
And share my screen. Can you confirm me if you can see?
Not yet, probably your desktop.
My presentation?
Yes.
Perfect. Thank you, everybody. I'm Mauro Petretta, Senior Scientific Advisor, and thank you all for being here. I would like to start this discussion on how bioprinting can change the future of healthcare by giving a definition of it. Several have been provided over the year, but all are based on the same basic concept, the ability to dispense with accurate spatial control, biological materials and living cells in the same process to create 3D structures. Bioprinting truly represented a revolution in the tissue engineering and regenerative medicine field because it enabled to pursue a more biomimetic approach to engineer complex tissue and at the same time target so far unsolved issues like vascularization and the regeneration of critical size defects through an automated and repeatable process that could be envisioned as scalable for clinical relevance. And that's due to its unique properties that bioprinting has known a constant increase in relevance in many different fields, starting, as I said, from tissue engineering and regenerative medicine, where it enables to create complex patient-specific structures in a highly automated process, to the drug discovery field, where it's used to implement robust, high-throughput testing platform in a highly biomimetic environment without concerns relating to tissue availability or ethical concerns for the use of animals in drug testing. And one of the field in which most recently its relevance is growing is personalized medicine, where it enables to create patient-specific medication regimes by creating a solid dosage form with tunable release profile and the possibility to combine multiple active compounds. And that's exactly to address all the complex needs of so many different groundbreaking application fields that we at Regeneration as pioneers in the field have been trying over the last 14 years to come up with solutions to support you through the whole biofabrication process by a combination of hardware with our RGen platforms and software with our shaper and controller. Our goal is to help researchers in each phase, from the preparation and design of their experiment and structures to the definition of the optimal protocol and printing parameters and execution of the process in the optimal conditions, to real-time monitoring and evaluation of the outcome of that process and the possibility to adapt it on the fly to correct eventual mistakes. Now, in order to give you a glimpse of all the potential that bioprinting has to offer in different application fields, I would like to make a quick overview of application made by our scientific partners all over the world. I would like to start with this one that saw us in first line cooperating with Utrecht Medical Center, where we show the possibility to combine very different processes as bioprinting and multi-electrowriting to take advantage of the unique properties of both. We show the possibility to perform the multi-electrowriting process on carved anatomically relevant surfaces in different materials such as hydrogel or bioceramics, and we use this to reinforce a solid hydrogel with a mesh of micrometric multi-electrowritten fibers in order to create a bioprinted condyle structure that was shown able to induce cartilage matrix formation up to 28 days of in vitro culture. Staying in the same field, the bone and cartilage regeneration, this study from Trinity College Dublin showed the possibility to combine multiple printing techniques in the same process to generate osteochondral implants. As we can see in this quick video, thermoplastic polymer was used to build a framework that was functionalized in the bone region with cell-laden hydrogels and sacrificial ink to promote vascularization, while a drop-on-demand technology was used to dispense in the cartilage region a highly concentrated suspension of chondrocyte and stem cells. In this way, the polymeric framework itself could work as a sort of microchambers in order to promote spheroid formation and maturation, and at the same time, upon polymer degradation, induce their fusion and the formation of new cartilage tissue, showing the possibility of fabricating human joint surfaces able to promote new tissue formation up to 10 weeks of in vitro culture in static and dynamic conditions. To a different anatomical field, this study from Tel Aviv University showed how it's possible to create bioprinted cardiac patches with built-in electrodes in a single process. As we can see in this video, the electrodes themselves are printed in a conductive ink and combined with insulating layers in PDMS and with patient-derived cell-laden hydrogel to form these flexible soft patches that were shown to be biocompatible and able to withstand physiological loads. Moreover, the embedded electrodes made it possible to monitor the engineered tissue function and control its contraction through external electrical stimulation. Of engineered tissue, skin is certainly one of the most appealing and researched one, and this study from National Institute of Health showed the possibility to combine 3 different dispensing technology with ad hoc sets of biomaterials and cell population in order to create a high throughput tissue model by replicating directly within well plate formats these models that showed to be able to replicate the native skin tissue structure and barrier function, and obtain a mature tissue model in less than 2 weeks. In a follow-up study by NIH, this bioprinted skin model was also used to test the efficacy of chemotherapeutics in a skin carcinoma tumor model that was achieved by using cancer spheroids to invade the normal skin constructs. This showed the possibility to use bioprinting to obtain clinically relevant model to test therapeutics drugs efficacy in a tissue that is recapitulating the native disease biology and that can be in the future envisioned to be used in combination with patient-derived cells to create targeted, personalized treatments. Last study I would like to present you from the University of Nottingham showed as a proof of concept the possibility to create a personalized tablet by combining 5 different active compounds with independently controlled release profiles by changing the architecture and the composition of different compartments in the tablet. In this way, it is possible to
Mauro,
obtain...
you have 7 minutes. Sorry.
No. I will close very fast. In this way, it's possible to create complex medication regime that are tailored for the individual patient needs. And with this, it will be all from me. I thank you all for your attention, and I invite you to contact us if you have any more questions about the bioprinting world through our email or social channels.
Thank you, Mauro, for this overview in printing. And now we continue with Ulf Hedler from EBD.
Yes, here we go. I was preparing my background screen that way, actually, that Rudolf is already in a perfect mood, actually, for using the Microsoft system directly on a beach. So you can see that. Nice background, which is unfortunately in Microsoft background, so it's not a picture that I've taken personally. But here we go with the talk that I'm going to give. And what I will introduce is a little journey from cell-based assays to more clinical relevant data. And it shows actually the development that we did from the very early days actually in EBD, where we did things like wound healing assays, and where we are now moving more to patient relevant systems like CAR T-cell assays that we are doing. The work that I'm presenting here is actually mainly done by Jan's group. He's heading the development team for our disposable group. And I'm one of the founders of EBD and taking care of everything that's needed. In principle, I think most of you know EBD for the labware that we are doing. But one point actually that Patrick mentioned very importantly is that you need to keep the physiological conditions that you have the cells in really on a high standard. So on the one hand, actually, it's the labware that we are doing that allows us to perfuse. So that's the reason that we do pump systems. And on the other hand, also we control, of course, temperature and humidity in the assays that we are doing. And lately, especially to host 3D assays, we started to have a small group of reagents where we also do things like collagen and things for staining, especially actin, to show the capability of the assays that we are doing. Now, in the past, EBD was really a company that was doing chambers for cell-based assays. So you see things like immunostaining. We do chambers for angiogenesis and for chemotaxis and migration, as well as perfusion. And as we all have a background in biophysics, you see that all of these chambers have a little trick to facilitate these assays, either to preventing, for instance, meniscus, or in these channel slides, especially to perfuse cells for long-term physiological conditions. Now, taking a picture after these assays is one of the things actually that you are initially trying to do, but to analyze these data and really have the capability to quantitatively analyze the results that you are going to have is one of the things that we have added lately to the company, and that really helps to standardize these assays, and that is a very critical point actually in these assays. Here you can see one of the early assays that we did actually for wound healing, where you have a small insert that separates two cell fractions from each other. These inserts are made from a silica, from a silicon material, and you can remove them. Then you end up having these two patches of cells. And what you can do with that is something that you can see in this video here that was taken from a group in Kobe that was published in Science roughly 10 years ago. You can see you have two patches of cells, and these are able to mingle into each other so they can form a tissue, and you can nicely analyze the proteins that are involved in these processes. And if you delete the proteins that are responsible for that, then you can see that this intermixing doesn't happen anymore. So that's a classical assay in 2D, and of course that brings a lot of impact in research. But how much does that really help a patient? And that is, of course, the discussion that we are having here. So the chambers that we are classically doing are consisting of two things. So the upper chamber that really gives you the design and the type of assay that you run. And then classically, you have a cover slip underneath to run the assay in a microscopical analysis fashion that you can really decently analyze these data. Classically, we are trying to immobilize the cells on the surface. So a functional surface that we are using is called EB-treat. So that allows us actually to host the cells on the surface. Lately, we have added actually a compound, a surface that is called Bioinert. And that is a hydrogel that prevents the cells from attaching to the surface. So what you can see that instead of adhering to the surface, now the cells start to cluster and to form aggregates and sit on the surface. Now, what a hydrogel is something that you can see on this picture. So instead of allowing a cell to anchor to a surface, now we are classically forming a water film. So that is what a hydrogel effectively is. And then these cells that are floating around on these surfaces, they can only anchor at the spots where we would like to have them anchored. So you see, actually, you have that bioinert surface that prevents any cell attachment. Then we can do chemistry on these surfaces, anchor a certain ligand onto that, and that would allow to host cells on these surfaces. And then you can make patterns like that one here, where you can see actually the name of us. So we have areas that prevent cell attachment, and then we have areas that allow cell attachment specifically. Now we can do that with multiple type of arrays, of course. We can define that in single cell arrays or in multi-cell arrays. And The assay that I would really like to show you is a cell assay here, where we can really do clinical relevant testings, because here you can see some renal cancer cells. And what we are trying to do is we are immobilizing them on a grid of these attachment spots. And now we can bring a T cell close to them. In that case, that's a CAR T cell that allows us to eat these type of cells up. So what you can see in this movie here, and that is a very important step here, we are using, of course, an artificial intelligence system to use that. And what you can see is this AI algorithm is screening where that pattern effectively is. So you can see in the first part here, everything is red, showing that the cells aren't locked in. And then actually the algorithm is looking around, recognizes where the cells are, and then you can see that now it recognizes that pattern, it overlays actually that pattern. And now, especially if you're looking here at that lower line, now you can see that we have recognized these renal cancer cells. We are adding T cells to that. Then you can see that now we are killing all these retinal cancer cells that are sitting on that very surface. You can see that we are removing all these cells here. So we are moving again from a red square, from a blue square where the cells have been sitting, to a red square where these cells have been removed. And that way
If
you
you
can
have
really
7
recognize.
minutes, sorry.
Yep. That way you can really recognize that we are eating up all these cells. In this last picture that I would like to show you here is that we cannot use that only for immobilized for cells that are initially immobilized, but we can use these bioinert areas also to allow spheroids to form directly under physiological conditions, so we can flow material over these. Then only these spots here would allow the attachment of spheroids on the flow, and that way we can really recognize spheroids and form them very well defined on an immobilized surface. And with that, I'm perfectly done in time. Thanks so much for.
Thank you very much for all the speakers. Very interesting, and apologize for having to stop you because you have a very, very interesting talks. We have a question from the audience to Mauro and Patrick. They say, in regards of tissue engineering for clinical application upscaling, it is considered a bottleneck. What is your view on the solutions to overcome these bottlenecks for bioprinting? They say, for example, size of construct, spheroid formation, etc. So
yeah. Patrick, if you like, I can start.
Yes,
And,
please. Just what I want to say. Yes, please, Mauro.
Absolutely. So certainly for the time being, bioprinting is mostly used in the research field, but for regulation reasons, that for technical limitation concerning scalability that are being addressed right now. But as I said, there are possible solutions and field in which this is already to a different level of maturity, like in the drug discovery field, and the possibility to have this process replicated in an automated manner really already is a very big first step for what concerns future clinical translation. For what concerns sides of the structure, there are application approaches that could help overcome printability issue, like the free-form fabrication methods, for example. But also it's important to focus, as we said, on vascularization and the ability to provide the nutrients to bigger structure in order to be able to create bigger constructs.
Thank you, Mauro. And yeah, I love your question. It's really important to see that the future operating room will be the laboratory. So there is this big shift of how will medicine be done. And that's exactly the point. We need to make science applicable. And if we look what is going on in science, there's clear everything is changing towards a wonderful and better future. I mean, it will reduce drastically suffering, and it has potential for global scale that everybody has access to regenerative medicine. So if you think you just need a lab, you need some cells, and some recipes to program these cells. So that is like everybody in the world has an iPhone today. It will happen with that as well. And regarding our contribution to that global movement, we just have seen from the clinical point that we were not able to do our transplantation work properly. And that's the reason why we developed our platform. So if you have any needs for regular spheroids that can be used for clinical purposes, get in touch with us. You can scale indefinitely to clinically relevant amounts. So 50 plates of the medical plate give you 1 million spheroids, and it will be a medical device, so it will be certified, so you can really prove it and work in the lab. And the beautiful thing is they are like building blocks, so you can put it into a 3D printer, and you can print also spheroids, which also physiologically is quite relevant because it has been shown that spheroids show some function already, which single cells often do not have, and you can sometimes be more effective being in the biofabrication field to create something meaningful. Yeah, that's my thing.
Thank you, Patrick.
I hope I answered the question right. Yeah.
Thank you, Patrick, and thank you, Mauro. I think you give interesting points of view from your own background and your own expertise in the company. We have here another question that is I think it is given to Ulf. They ask if you could provide examples of cells that have been analyzed using this invasion assay that you presented.
Yes, there have been a ton of different cells, of course, that have been tested with that. Many of that have been either cancer cells to test invasiveness. There have been mixtures of cancer cells and endothelial cells to form really or to learn more about the angiogenesis side of that. And then there's also a big group of keratinocyte assays that have been done to really look at classical wound healing. There have been tests with viruses, for instance, where people added viruses and see how that changes the migration of these cells. So there is really a ton of publications. We have a very good search integrated on the EBD website. So if you're looking for more details there, Sinetra, you will be able to find that definitely in more details on the website, or you can address me, of course, personally as well on my email address.
Thank you, Ulf. And I want to also give a question to Vincent. What are the plans for this program, Tools for Life Science, for the future? What is your highlight for the plans in regenerative medicine?
In the field of regenerative medicines, we see an increasing request from going from manual workflows to automated workflows. And we were able also, as I was showing, to develop some building blocks around these technologies. But to really see again this in regenerative medicine, the isolation of the cells that you want to make that automated, the expansion and the engineering of the tissue at the end. And then this is really where we concentrate, and we, you would like ideally, of course, to do all these steps within a fully closed system with as little interface as possible. The reality shows that, of course, you have to work in a modular way, but this is, of course, the vision that is driving us.
Thank you very much. If you allow me, there is one question. If you can, Ulf, help us to reply to this question very fast. They are asking, is the deep learning optimized for one single cell type, for example, the cancer cell, or does it need to be optimized for each intended tumor type?
In principle, it's not. It's really a recognition of a certain pattern at the end of the day. But of course, it depends a bit, as we are using phase contrast initially to run these type of assays. It's always a question whether the algorithm recognize these type of cells in that same phase pattern as it does. But it should, in principle, allow most tumor cells actually to be recognized and standardized, but might need little optimization, but it shouldn't.
Thank you, and I thank to all the speakers. It was very interesting insights of the emerging technologies, such as the 3D bioprinting, microfluidics, these interesting organ-on-chip or cell-on-chip assays. Therefore, let us present you a multidisciplinary group, which bring us to the table some of those technologies through an EU-funded project called OrganTrans. And also we have a talk that will help us to understand how it is enabled the EU funding for research organization and company in generative medicine field done by AMIRES. So for no further introduction, so I will give the word to Gil Vera, who will talk us about OrganTrans project.
Thank you, Mariana. You see the slide?
Yes, we can see it.
Perfect. So from lab to OrganTrans and from OrganTrans to clinic, as project coordinator, it's my pleasure to present you OrganTrans technology, but more specifically the alliance behind this RegMed project. Tissue engineering is the construction of bioartificial tissue. You need cells, biocompatible scaffold material, and an assembling system directly connected to a maturation environment. The vascularization is a worldwide challenge in regenerative medicine. You could look at the vascular tissue engineering challenge published by the NASA. It won't be fully solved in OrganTrans, but we will contribute. Standardization is also required from the concept phase to develop systems that are compatible with GMP and ATMP requirements. You see now all the partners, including CSEM as technology orchestrators and AMIRES as partnership orchestrator. The most common model organism that are used in biomedical research are Caenorhabditis, Drosophila, Danio, and mice. This model, as well as 2D cell culture and human organoid, are assessed for their relative benefits and limitations. Organoids are not only strong candidates for drug testing, but they also represent the building block for organ replacement. And the overall concept of OrganTrans is to combine organoid technology and mimicking tissue architecture for liver. The autonomous self-assembly uses autologous cells from residual healthy tissue, and the biomimicry is achieved by bioprinting using synthetic hydrogel and endothelial cell for vascularization. So the goal of OrganTrans is to develop the technological blocks for organoid transplantation with a specific target for patients with chronic end-stage liver disease having still some healthy tissue. The process flow starts with the isolation and expansion of adult stem cells, followed by the standardized production of spheroid using supporting cells composed on bone marrow mesenchymal stem cells and endothelial cells. To avoid uncontrolled proliferation and uncontrolled cell differentiation, the spheroids are handled and sorted according to strict criteria. In addition to spheroid, synthetic hydrogel, endothelial cell and mural cells are loaded in the bioprinter to manufacture a complex vascular network. And finally, the engineered bioconstruct is then perfused and matured with the in vitro testing before in vivo testing in immunosuppressed mice. Finally, it will be maybe deployed under the compassionate use exemption with 3 leading European transplant centers belonging to the consortium or to our advisory board. We touch now really a crucial point, that is the alliance. And I am very proud to have research institutions, technology providers, tools providers, patient associations, therapeutic developers, and medical centers with the King's College and the University Hospital from Heidelberg and Zurich. And all together, we can deliver building block and technological block serving regenerative medicine while keeping the balance between creativity and regulation. It's crucial to have dedicated program for applied research, for innovation, and with the constraint of regulation. So at all attendees, please contact this strong alliance to share new ideas of collaboration. And now I pass the words to AMIRES, the alliance orchestrators, to give you more details. Thank you.
Thank you, Gio. And from AMIRES to AMIRES, I give the word to Dr. Rudolf Fritzek. He will talk us more about the EU funding opportunities.
Thank you very much. Thanks, Mariana. Thanks, Jill. So not yet on the beach. Very much amazed by all the presentations done so far. So let's have a look in this. I'm not going to show you these beautiful slides. I will show you very functional texts of Horizon Europe calls. And okay, so Mariana, can you confirm you see the slides?
Yes, we can see.
Perfect. Thank you very much. So, okay, just a few words about AMIRES. We are the supporters of getting the consortia together and getting funding. So we are very much involved in projects with the business or strong clinical impact. And indeed, we believe that we are this partnership orchestrator, as it was highlighted several times. We are relatively successful. We were relatively successful in Horizon 2020. We hope that this will be also the situation in Horizon Europe, which is about to start. You probably realized that yesterday there was an approval from the members of the European Parliament about the final budget. So this is now getting to be reality. So there's 95.5 billion available for next 6-7 years. And now actually the calls are almost ready to be deployed. I will not speak about all Horizon Europe because I would need several hours for this, but indeed the regenerative medicine will be mainly in this, can be found in Pillar 1, where there is this excellent science. It will be also certainly in the Pillar 3, where we see this European Innovation Council and with Pathfinder and transitions. There are even specific calls related to RegMed. And I will speak mainly about the opportunities in Pillar 2. There is this type of collaborative project you just seen in OrganTrans, and I will particularly focus on few topics in health and this digital industry and space clusters. First topic I would like to put into your attention is this 2021 call on next generation of advanced therapies to treat highly prevalent and high burden diseases with unmet needs. And here, clearly, it calls for new advanced therapies based on pluripotent stem cells, gene editing, RNAs, and they will be pushed through the clinical trials. The projects are going to, they are particularly requesting the preclinical research platforms, and some examples are clearly described in this WHO report on the priority medicine. So the typical diseases would be chronic non-communicable diseases, heart diseases, strokes, osteoarthritis, Alzheimer, hearing loss, alcohol liver disease, chronic obstructive pulmonary disease, and so on and so forth. The idea is to have these advanced therapies tested in vivo and to have them ideally on the market within the next decade. The project will have roughly the budget around 6 million, and the budget is relatively generous. We are having 60 million for the overall call, so we assume that there will be a lot of demand for this one. Please understand that I'm presenting the draft, so always consult the latest and the final version. The next topic, which may be linked to RegMed, is this one on the smart medical devices and their surgical implantation for use in resource-constrained settings. Here we speak more about smart, sustainable and affordable smart active implants but also on the surgical procedures of these implants, which would ideally be minimally implanted with minimal invasion, and it should be validated through the clinical trials. So here, it's what we mean, or by what Commission means by these smart active implants. It will be a microelectronic component, either sensor or actuator or similar. There's a strong need on the an application in low-income countries, so the cost-benefit analysis will be certainly helpful there. And here at the bottom, there are some examples of diseases, either orthopedic, neural, cardiovascular, or metabolic. So therefore, this means that it will somehow link the typical examples like pacemakers, in vivo biosensors, cochlear implants, deep brain stimulation. This project is an innovation action, and the budget should be around 6 million as well, the EU contribution. The overall topic budget is a little bit smaller. It's not typically the RegMed, but I imagine that the biointegration will be a very important aspect for these smart medical devices. Therefore, the structuring of the surface or some kind of response stimuli. It can be also the EBD technology, which may play a role there, or even some kind of a drug-eluting functionality. These 2021 calls, they are expected to have a deadline at the end of September 2021. The 2022 health course, they are expected to have a deadline. There should be 2 stages probably. The first stage deadline will be in February, and the next stage will be September 2022. A little bit into the third topic. So we have a preclinical development of next generation of immunotherapies. And indeed, here the main idea would be to restore the ability of the immune system with antibody-based, RNA-based, or cell-based therapies. Here explicitly mentioning health technology assessment has to be there at the start of the proposal. It is not only health cluster where we can expect projects linked to regenerative medicine being applied. There is also a couple of topics in the digital cluster, where, for example, here this is a perfect topic for the development of technology devices for biointelligent manufacturing, where we are trying to, or where the Commission is trying to call for a cooperation between key-enabling technologies using biological system and interfaces making decision. The wording is relatively complicated, but we can imagine, for example, having a 3D printer linked directly to bioreactors and having a clear feedback loop between these two manufacturing technologies. There is also a very nice call in 2022 in this program calling for smart and multifunctional biomaterials for health innovations. There, the application will be somewhere between advanced therapies, medicinal products, or medical devices. And explicitly, you see the wording asking for a tissue engineering, artificial organs, implants, microfluidics, bioactive scaffolds, and so on and so forth. Also here, the preclinical regulatory will be important, also including the upscaling and good manufacturing practice, similar to what we have right now in OrganTrans, and present solid economical evaluation will be very important here as well. With this, I would like just to conclude that you see that we are in a transition in Europe to the value-based healthcare, and with the wording of the calls, the cost-benefit analysis or health technological assessment, this economical assessments before actually deploying the larger clinical studies are very important. Therefore, we also associated with this company called Bettera, which can do this type of studies, of course, bilaterally, but also within the projects. And with this, I would like to conclude. AMIRES, of course, will be happy to support you with the proposal preparation, with contacting between different partners, between different bodies, and here are just the contacts to me and to Mariana. Mariana, Jill, back to you. Thank you very much.
Thank you, Rudolf. Thank you, Jill, for this presentation. So this time we will jump directly to the next session. We will direct the questions to the speakers. For that, let's start this last session to clinic, and we can hear about the clinical applications of innovative solutions. Therefore, it's my pleasure to introduce you to the first speaker, Martin Pravda from ContiPro. So the floor is yours. We cannot hear you.
But
yes, perfect. Oh no, we cannot hear you again.
You are muted.
yes, is it better now?
Yes, we can hear you.
Sorry for problems. Now I have another question. Can you see my presentation? Can you see my presentation now?
It is still, yes, it seems so. Yes, now we can see it.
Okay, great. So, at first place, thank you for invitation. My name is Martin Pravda. I'm working in Research Center of ContiPro as a group leader of group Regenerative Medicine, and I would like to introduce you our activities in the field of development of new technologies for regenerative medicine. ContiPro is a Czech biotechnological company which is placed in the Eastern Bohemia. Our company was established 30 years ago, and our main activities are focused on the production of raw materials for pharmaceutical and cosmetic industry. Besides that, ContiPro runs a huge research center which is focused on the development of new technologies for innovative therapeutic procedures. As I said, our main interest is the production of raw materials for pharmaceutical industry, especially the hyaluronic acid. Hyaluronic acid is a polysaccharide, polyanionic polysaccharide from the group of glycosaminoglycan. The most important thing is that hyaluronic acid is an integral component of mammals' tissues, so that's why it exhibits excellent biocompatibility and biodegradability, and that's why it found during last decades many different applications in the field of the wound healing, ophthalmology, and development of products for viscosupplementation. The basic function of hyaluronic acid in the tissue, we can divide it in three main roles. It's a hydration of the tissue, and hyaluronic acid, thanks to its physical chemical properties, provides to tissue the mechanical strength. The second important function is control of ion exchange and diffusion of biomolecules through the structure of tissue. And last but not least, hyaluronic acid is capable to interact with the different kind of cells receptors, and that's why it is involved in the regulation of cell cycle. Because of these properties, hyaluronic acid was proposed as an ideal material for development of bioinstructive materials, which can control the cell proliferation, differentiation, and motility. And of course, it also can be used for production of materials for control release of different biological active compounds. Here in ContiPro, we are able to process hyaluronic acid in many different forms. We are not interested just in the production of the hyaluronic itself, but also we are able to provide different kind of materials like thin films, microfibers, nanofibers, hydrogels. So these materials can be further used for development of medical devices or different more complicated medicine product. The One problem of biomaterials which are based on the natural hyaluronic acid is their short biological half-life and poor mechanical strength. This problem could be overcome by chemical modification of hyaluronic acid. So here in ContiPro, we are interested in the development of new chemical procedures which can lead to the hyaluronic acid derivatives, which provides extended biological half-life, better mechanical properties, and they allow us to encapsulate the cells in their structure and so on. Of course, that any new chemical composition of the material brings the issues with its safety. So one of our main activity is to prove that the materials which we developed are safe and functional. The most common way of modification of hyaluronic acid is cross-linking of hyaluronic acid molecules. This procedure, if we perform it in a water environment or in a aqueous solution, leads to the formation of covalently cross-linked hydrogels, and these hydrogels provides better mechanical strength of these materials and extended biological half-life. For the field of regenerative medicine, it is very convenient to have a system where this cross-linking reaction proceeds under physiological conditions, in the presence of the cells or in the contact with the living tissue. In ContiPro, you develop several derivatives which provides these properties. Here you can see the structure of the hyaluronic acid tyramine derivative. This derivative could be cross-linked by enzymatical process, which is mediated by horseradish peroxidase, or by photocross-linking reaction. This derivative is at this time produced in a pilot plant scale. We are using Cgmp process, and different materials based on this derivative were already evaluated by different safety protocols, included the ISO 10993 procedures. So we can say that this material is safe and is suitable for use in clinical trials.
Martin, you have 30 more seconds.
Okay, so these raw materials serve to preparation of hydrogel precursor solution, which can be cross-linked during, for example, surgical procedure, and provides materials with tunable mechanical strength, gelation time, and swelling properties. Our materials were used in many in vivo and in vitro studies. We are able to combine them with different kind of the cells and biological material like blood products, for example. And to summarize, the main activities of the ContiPro is the development of original hyaluronic acid derivatives and materials based on these derivatives. We are able to produce these materials in pilot plant scale, and these materials should be suitable for further development of medical devices and other medicinal products. So thank you for your attention.
Thank you, Martin. So the next speaker, we have Christian Hirst from a spin plant in Germany. The floor is yours, Christian.
So hello to everybody, and thanks a lot for the invitation. I hope you can see the presentation. and I'm happy to present SpinPlant. We are a very small R&D company located in Halle in Leipzig in former Eastern Germany, and we are concentrating on electrospinning. We are electrospinning collagen, and why do we do this? We do this to create a tissue that is as close as possible to the natural gold standard. When we talk about biomaterials, we talk about mimicking nature, we talk about mimicking the autologous gold standard. And we are trying to do this in the field of bone, cartilage, and skin. And when you take a look at the extracellular matrix in these areas, you find content and structure. When I talk about content, I talk about native collagen. And when I talk about structure, I talk about nano and microporous three-dimensional scaffolds. So we are trying to mimic just that. We are trying to have a native collagen content that is bioactive on one side, and we are trying to create a scaffold that is nano, microporous, and three-dimensional at the same time. And important, it should be stable in vivo as well. The structure should remain the same in vivo as well, because only then cell adhesion, cell migration, and cell regeneration takes place. So how are we doing this? We are creating a spinnable solution that consists of native collagen. We are then electrospinning that spinnable solution. We are creating electromagnetic field, and by having a carrier on the other side that is picking up the nanofibers, we create specific thicknesses, specific sizes, and we're creating a nano and a microporous structure. And we did that since 2014 at the Fraunhofer Institute in Halle. And I can tell you it was quite a job. It was quite a job to really create a spinnable solution. It was quite a job to find native collagen that is working. It was quite a job to optimize a scalable production process. And it was quite a job to stabilize the collagen afterwards to have it really stable in vivo, which is, as I said, of high importance. But we managed to do so, and therefore now have 3 products that are developed. And these 3 products that are developed are in the field of bone healing, in the field of bone filling, and in the field of cartilage repair. So we are concentrating on advanced bone healing. We are concentrating on orthopedics, trauma surgery, and dental applications. And with regards to spin base, we are providing a tissue engineering scaffold to be combined with other components like growth factors, MSCs, or BMPs. You can see how our products work. Spin Heal will work to really cover the bones, diabetic bones and burn bones. Spin Fill can be used to fill the bone voids, and you see here the filling of a cage for spinal surgery, and Spin Base is to be combined with other components. To give you an idea, for Spin Fill, for instance, we are co-spinning nanoparticles of hydroxyapatite, and therefore we can enhance bone formation and create osteostimulation. And we could do that with growth factors for bone healing at the same time, but it's working without these further components as well. We did a lot of testing with competing products at the same time, and you can see that we did quite well. You can always see that the structure makes a difference. The competitor that is shown below, you can see that the structure is not stable in vivo. And as you can imagine, for cell migration and adhesion, that is really making the difference. So it's not only the bioactivity of the collagen, it's also the structure and the stability of the collagen. I don't need to tell you, making a medical product is a big job. So we are now in the phase that our chemical characterization is done, our prototypes are done. We are certified according to 13485, which is quite a job. We have the quality management system in place. We are facing the third recertification in June, and we can't wait, as you can imagine. It's terrible. They are taking you for 3 days and going to the quality management system. And so we are all done with all the in vitro testing by now. And what follows to receive the 510(k) approval is a feasibility animal test. And we need that for the CE certification at the same time. And we start the approval in the US because we can do it without clinical evaluation by using a predicate. And after we did that, we follow with clinical evaluation for the approval according to CE. So we hope to get the approval next year in the US, and the year after in Europe, which is kind of sad because we are a European company, but we are more or less forced to start in the US because clinical evaluation, as you can imagine, is very cost intensive, and we're in the middle of a financing round currently to get the money to get that all financed. So that is more or less the story about Spin Plant. Happy to discuss with you further questions at a later stage. That's where we stand currently, and we, as you can imagine, are now looking forward to get our market approval. That is a big goal we are going for. Thanks a lot.
Thank you, Christian. Thank you for the talk. And our third speaker is Hanna Patochikova from Bioinert in the Czech Republic. So, Hanna, the floor is yours.
Thank you for the word. I'll try to share my slides. Oh, can you see it?
Not yet.
Okay.
Are you sharing already, Hanna?
Yeah, I think so. Yeah. Wow. It's just the icon just next to the leaf.
I can support you with the presentation.
Oh, yeah.
Because we cannot see.
Mm-hmm. Well, if you still can't see that, that would be great. Because, well, yeah. Well, actually, I don't see anything else I could do because it writes that I'm sharing, I think.
Okay, sorry for this.
Well, oh, yeah. Is it better now?
No, we cannot see anything that you want to share. Oh, you're sharing now. Perfect.
Okay, perfect. So
Oh, but I'm sorry, I have to stop sharing. It is a bit, there is a delay in the sharing system. I'm sorry, can you start sharing again? Please, sorry.
Okay, no problem. Okay, so
yes, perfect. It's a bit delayed. Perfect.
okay. So good morning to all participants. My name is Hanna Patochikova and I work at the Department of Clinical Research at Bioinert. And today I'm going to present our most advanced RegMed project that we have. So, Bioinert is an academic spin-off. It was founded in 2008. It is based in Prague, still in the academic compound, but it operates independently. We have our own funding, own staff, and own equipment. Bioinert is quite a small company with less than 20 full-time employees. We have quite a broad range of activities. We produce medical devices, we work as an analytical laboratory, but our core activity is manufacturing and testing of stem cell-based medicinal products. These are called advanced therapy medicinal products, and more precisely, our products fall into the category of tissue engineering products. Currently, we have two products in our pipeline. The first one is for the treatment of cartilage defects. This is under clinical testing, and the other one is for treatment of retinal diseases. This one has passed preclinical testing. Well, what needs to be done if you want to develop a TMP product? We began with laboratory research. This is what we do in-house. Currently, we focus on the robustness of the manufacturing process. Our products are autologous. That means that we deal with different donors, with different medical history, different age, and we need to be sure that we'll be able to deliver the product within specification, regardless of the donor variables. The next recent task was antibiotics-free manufacturing. The regulatory body wanted us to do so, so we had to switch to antibiotics-free production. And for practical reasons, we still work on the increased shelf life of our products. Currently, we guarantee 72 hours in a refrigerator. The next step was manufacturing. The product has to be manufactured under our GMP rules. We have all the certificates from the Czech drug agency. The most difficult task was aseptic manufacturing. Our products have to be free of all pathogens, and therefore they are manufactured in clean rooms class A and B. The next phase is testing. When it comes to preclinical testing, we outsource all animal tests to a GLP laboratory. If possible, we try to replace animal tests by in vitro tests, or we use the evaluation of published scientific data. And the clinical phase is done in-house. I mean, our clinical trials are sponsored and organized by Bioinert, and they are conducted in large hospitals in Czech Republic. Currently, we have two ongoing trials. Both of them focus on the treatment of knee cartilage defects. Phase 1 trial is a safety trial. It's monocentric, quite small, and it is in its final stages at the moment. Phase 2 trial is efficacy trial. This one is multicentric, randomized, controlled, and it has just begun. As you can see, there is an overlap between these two trials. That's because our regulatory body allowed us to begin phase 2 based on the interim data of phase 1, which slightly sped up the whole process. And here are some brief results of the safety trial. It has a standard design. Safety is evaluated as an assessment of adverse events, their relationship to the treatment, their severity, seriousness, and so on. And we also have some preliminary efficacy data. These are results of questionnaires. Patients generally reported improved knee function. And this data was supported by imaging methods like X-ray and magnetic resonance. However, it needs to be noted that these results are from a small uncontrolled trial and have to be confirmed in a larger controlled efficacy trial. Okay. This was all from my side. My take-home message would be that the development, manufacturing, and early clinical testing doesn't require a large team, and it can be done by a very small company if you have the right people and enough courage.
Thank you, Hannah. We will keep the questions at the end of the session. Therefore, I will give the word to the next speaker. Thank you, Hannah, again. To Vincent Ronfard from Cutis company from Switzerland. So the floor is yours, Vincent, and we're happy to be you're here.
Yeah, do you see my screen?
Yes, we can.
Okay, so I'm working for Cutis. I'm a Chief Innovation Officer. I put the frame of Patrick to make him happy. What we do is really our old technology is autologous cellular therapy. So and why we do that, because we are all made of cells, and we learn from the amplification of cells from the beginning, and this is tissue engineering. Our body is tissue engineering, and also we made our first scar just after birth from the nombril. The human body is the best bioreactor, and there are replacement of cells. So we have a lot to learn when we do tissue engineering from the clinical. From the biology of the human being. The problem is that we are very bad at to regenerate ourselves and to repair ourselves. If you look at human skin, we see that human skin has to fully regenerate about every month. And every month we have new cells. It means that a person today and 1 month from now would be the same person, but would have a different skin or different cells, which is very important, because when we do tissue engineering, you need to preserve this quality, which means that you need to have stem cell in the tissue engineering. There is a huge need for skin repair, and that's why we do this work. And as you can see, there is about 50 million of skin defects in the world. And it could be done from burns or for reconstructive surgery for chronic disease. But tissue engineering has been developed about 40 years ago, and ever since, this time, this is still the same type of concept and technologies we are still using in tissue engineering, at least for skin. And this work was coming from Boston, Harvard and MIT, and that gave rise to 5 different companies that still exist now. And the technology we are using at Cutis inspire from one of these technology. As it was said in previous talk before, there is a big challenge with tissue therapy and cell amplification. It's due to the nature of the biology of the cells. Cells are plastic. It means that they react to environment, so it's a moving target, and it's not easy sometimes to manage, and particularly with the process. So we want to make sure you always look at the biological property of your cells during your process. But if we want to do tissue therapy for use on human being, we need to scale it, we need to automate it, and we need to use a closed system to make sure the aseptic chain is still properly done. Definitely also the cost at the end is very important, because if you have a technology that is great but is so costly that nobody can afford it, it's useless. Also, the product needs to be reimbursable, so it means that very early on also you have to understand how you're going to finance your product. But finally, cells are not like drug, and in terms of regulatory, that's very important, because regulatory body now are much more aware of ATMP and biology, but they are still using a drug as a template, I would say, and it's not always suitable for cell product. What we use at Q63 the power stem cell, and we want to preserve this stemness. So if you can see here as an example, keratinocytes at the very tip of a hair follicle. When you isolate them, you grow them, they can make a 1 centimeter diameter epithelial autograft, which is show you that from few cells you are able not only to have something alive, but to make a meaningful epidermis. We use another setup, which is a tissue builder, the fibroblast, and you can see it can build tissue in laboratory. And this is our process. So basically, we take a small skin biopsy from an individual, we separate dermis and epidermis, we isolate keratinocyte, fibroblasts, we put them in collagen, and we get a tissue. These take about 30 days. And this is a product on the left, lifted from the tissue graft that was developed in collaboration with CSEM. And then on the right, this is an histology, and you could see the dermis, the collagen, the fibroblasts within the collagen, and overlaid with epithelial cells. We are currently in clinical trial phase 2, and in adult child for burn indication and also reconstructive. And you can see on the left, this is the current, the standard, the gold standard, which is the autograft. And then on the right, this is the novo skin. And you can see that the aspect is much better. So we will go to automation of the novo skin, and I explain the reason why, and if you want to have that available for the patient and reduce the cost. So this is the overall different step that we have in automation, from the biopsy harvesting to the tissue formation. And we are currently, this is the status of development we are now. We are trying to develop equipment for the different steps. So basically, we translated all of the manual steps into an equipment. And after that, we want to incorporate all of them together in maybe one machine that will be able to produce skin in about 20 days. This is done with collaboration with Zulke, because we need engineering, and also CSEM for the same reason. And hopefully we can continue this collaboration. When you develop something like that, at the end also, we would like to have some return on investment. So you want to position your product where there is a niche for it, and it's going to be used, and that's the case with cuties compared to other type of products. So we are a kind of unique in this regard. And what is the future? So I'm trying to brush that rapidly. The future is that, yes, we will need more tissue engineering, because if you look here between 2015 and 2050, the age population over 60 is increasing drastically in the world. It's also a very good time to develop tissue engineering and skin engineering therapy, because the technology is now at maturity. We've seen electrospinning, we've seen bioprinting, developing of more understanding of the biology using new equipment. It's now available, affordable, so the technology can really speed up and develop. And you can see from 75 to 2021, it took some time just for tissue engineering to be adopted. It went like waves. It was really developed, then there was some disillusion, then it was resurrection, that disillusion again, and I think the technology now is really at maturity. In the future, one of the problems with cells is that you need to use autologous cells to have a permanent take of these cells on an individual, but people are working to develop universal cells. Then when they can engineer cells to be developed by, used by everybody, and I think that will be maybe one of the good development for tissue engineering development, use as therapy for more people. In the next development, yeah, I'm almost finished. In the next development, you can also induce a diagnostic tool into your dressing or into your bioengineering skin. And the last one is to have a fully synthetic skin. That's very interesting. People are also looking at this aspect, but this is not what we do. Okay.
Perfect. Perfect timing. Thank you, Vincent. And we want to conclude with the last speaker, with Marcia Bedoni from the Don Carlo Gnocchi Foundation in Italy. So Marcia, the floor is yours.
Thank you. Now I'm trying to share my screen. Okay. Okay. Did you see my, do you see my screen?
Yeah.
Yes.
Okay, perfect. Good morning. Today I will talk about our regenerative rehabilitation approach thanks to extracellular vesicles in a specific case of cerebrovascular disease. First of all, a brief overview about extracellular vesicles that are natural nanoparticles deriving from different types of cells. There are different sizes of extracellular vesicles, exosomes that are the smallest one, microvesicles, and apoptotic bodies. All these extracellular vesicles are enriched in specific lipids, protein, and nucleic acid, and are present in several biological fluids. There are different functions about extracellular vesicles. In particular, they have the function of intercellular communication, and they induce signals via receptor and ligand interaction. In the last year, we are studying these extracellular vesicles derived by brain, and we study in peripheral blood as biomarkers for neurological and cerebrovascular disease. In particular, because extracellular vesicles are able to give an idea about patient stratification and to monitor the effect of rehabilitative treatment and regeneration processes. Did you see, sorry, did you see all the screen?
No.
For some, no, we see half of it.
Okay, sorry. I will try to change the screen. I don't know why, but my colleague said that she's not able to see all the screen. Okay.
Now, yes.
Now?
Oh, it goes again. At the beginning, it's fine, but afterwards,
okay. Not yet. Okay. Because I have a double screen, probably this is the point. And now? No.
We see it as a presenter view, but I can try also to share your presentation, maybe.
I'm sorry. So maybe why? Because yesterday is and now?
Now it looks good.
It's good? Okay,
yes.
perfect.
Yes.
Sorry. Okay, so let's start. Okay, about this, I would talk about today about this clinical case. That is the stroke, a cerebrovascular disease. Stroke is the second leading cause of death in the world, and the patient could have a spontaneous recovery or a rehabilitation recovery that would need to be hospitalized. In the first case of spontaneous recovery, we have two phases: the restoration of functionality of the brain and reorganization of the neural tissues. In the few days that both phases occurs, and there is a generation of different extracellular vesicles deriving from different cells of neural tissues. In the second type of rehabilitation, the patients, if a real severe effects occurs after stroke events, the patients needs a rehabilitation recovery with different type of rehabilitation, cognitive or motor rehabilitation. In all these cases of rehabilitation, there was a production of extracellular vesicles, in particular from brain and muscle tissues. I would focus about regenerative rehabilitation because regenerative rehabilitation is a new study in the middle between regenerative medicine and the traditional rehabilitation, with the main goal of to regenerate the tissue, to restore the functionality and to the social reintegration. Our focus is on this regenerative rehabilitation. There are many factors that influence the speed of recovery after stroke from the patient, in particular about the extension of the brain lesion or the patient age. This is a flow chart about the normal clinician approach. And first of all, there is a identification of patient's need, the clinical goal, intervention, and so on. And the regenerative rehabilitation biomarkers, names Rehabilomics, acts before the intervention to help the clinician to define the right clinical goal and the right final outcome, clinical outcome. We focused in our experimental setup 2 or 3 weeks after the acute event of ischemic stroke, where there is a global inflammation in a systemic inflammation, but in particular on the brain. We evaluate by clinical scale and by our biophotonics analysis. We isolate extracellular vesicle by size exclusion chromatography. We characterized by transmission electron microscopy. And then we introduce our characterization by surface plasmon resonance imaging. We first of all functionalized the SPR chip, that is a gold nano chip with a particular chemistry. And then we inject the extracellular vesicle and we perform a primary and a secondary characterization, also using gold nanoparticles to enhance the signals. I would show you only few results about this approach. We can observe on the left of the slides a typical sensogram that we can obtain taking into account that we are using a multiplexing analysis. We can observe different extracellular vesicles, different biomarkers deriving from all the type of neural cells derived from peripheral blood. And we can observe on the right that there is, in particular, an implementation in the GLAST that is a biomarkers related to neuroplasticity and neuronal damage, obviously in stroke patient. And also CD106, that is a typical biomarkers of endothelial cells, underline that is a remodeling of the vascular tissue. About the secondary characterization, we want to focus about angiogenesis and neuroinflammation. We can observe that in the first phase, 2 or 3 weeks after acute stroke event, there is not yet a real angiogenesis process started. But TSPO, that is a biomarker typical of neuroinflammation and neuromodeling, we have an increase, an overexpression in stroke patients. We also observe the same patients before and after rehabilitation, and we can observe that both in cases of angiogenesis and neuroregenerative tissue, we have a regeneration of the tissue post-rehabilitation, meaning that the rehabilitation treatment is obtaining the right final goal. We also compare our data, our experimental data about extracellular vesicle with the clinical scale used by our neurologist, and we can observe that it is a recovery after stroke about neuroinflammation and neuroplasticity. So concluding, as a take-home message, we can observe that extracellular vesicles can be used as biomarkers of regenerative rehabilitation, in particular, as in our case, on cerebrovascular disease, and also that our SPR biophotonics-based platform could be useful also to reveal extracellular vesicles related biomarker monitoring. Thank you for the auditors, and thank to my researcher teams. And also I would invite you to join us in Regenerative Rehabilitation Symposium. Thank you.
Thank you, Marcia, for the talk, and thank you all the speakers, all the experts on regenerative medicine for this session. So now we have still 15 minutes for question. So I propose you that we start with specific question and then with more global question. We won't be able to address all the question, but we will also then connect the different people according to the different question. So let's start with a question for Martin at ContiPro to know if the cross-linked hyaluronic acid can be controlled in terms of degradation, typically with enzyme or hydrolysis.
Yes, in general, it is possible. Those hydrogel are tunable in this way. For example, the degree of the cross-linking influence the rate of the biodegradation in the organism. Those materials are biodegradable by hyaluronidase, what is standard enzyme for degradation of these materials, both in vitro and in vivo. And also they could be degraded by reactive oxygen species. So it is one part of our research to prepare hydrogels with tunable time of biodegradability.
Okay, thank you. And quickly, can you comment and can you about the hyaluronic acid derivative, the TA, the tyramine that you mentioned, if you have some clinical application as well?
We are not at the clinical stage yet. As I said, we have several products in the pipeline which were evaluated preclinically. It means according to ISO, for example, and now for one product we are preparing clinical study.
Perfect, thank you. Let's move to Christian from SpinPlan. If you can comment, in fact, the range of stiffness of the scaffold that you can achieve, and also mentions the source of your collagen, please.
We're using bovine collagen class 1. We're using sources in the US, but we are cooperating with different suppliers, and we are working with bovine collagen from Europe as well, but we're facing regulatory problems when we're using European bovine collagen. Therefore, US concentration, at least for the US approval, is crucial. We can double and 3 times grade the thickness. I have a legal background, to tell you the truth. I'm the CEO, so with specific technical questions, I would need to send you to Javorina. She's head of R&D in our company, but we can double the size. By the way, as fast as the carrier material is moving, or the slower the carrier material is moving, the thicker the material becomes. So we can create really, differences between 100, 150, 200%.
Okay, thank you, Christian. Then to Hannah from BioInnova. So Matund is asking the working principle behind the BioCure for your cartilage defect, and in addition, if you have some need of ISO certification with 13485.
Well, yeah, I'll begin with ISO. Our product is a medicinal product, so we don't need ISO for medical devices. However, our product is a combined product. Our stem cells are loaded on a scaffold. This scaffold is commercially available. It has a CE mark, and the stem cells which are on the scaffold are inserted into the cartilage defect, and in there they are attached to this scaffold in a patented manner. Let's say we use autologous blood plasma, and so these stem cells, they remain in the defect, and they help the cartilage to regenerate. There are biologically there are two main principles. Firstly, the cells stem cells can themselves divide and possibly differentiate into cartilage cells. But more importantly, stem cells release growth factors, and they help other cells to regenerate and to form the cartilage.
Okay, thank you for the answer. So now we have several questions for Vincent that all goes in the same direction about the functionality of the tissue and the sweet gland or any type of gland with the engineered tissue. Can you comment if you have the functionality from the original site, or are you going to have some hair that will grow at the final location after transplantation? You are muted, Vincent.
So it's better with microphone, sorry. Yes, in terms of the functionality, in our case for de novo skin, we are using two cell types, keratinocytes and fibroblasts. They are autologous, so they're going to be on site permanently, hopefully. And we have, so the fibroblasts, we help to rebuild the extracellular matrix and the dermis, and it will allow ingrowth of blood vessels and repopulation to regenerate a normal tissue. For the epidermis, we are using keratinocytes, and there is no regeneration of sebaceous gland hair follicle, and it's not really occurring in any type of tissue engineering. Hair follicle regeneration is quite complex. Sebaceous gland too. I mean, there are progress made with that, but it's not occurring with our product right now.
Okay.
So yeah, does it answer this?
Yes,
okay.
And there is also a demand if it's possible to buy some skin sample, typically for experiment or any testing on hair. Or...
Yes, so we the product for clinical use is not available, obviously, because we are in phase 2, pivotal trial. For testing and research, that's something we can discuss on case by case, because that's not what we are doing on a regular basis, and we have to see if it really fits into the need of the person what is looking for, because it's a big size, so you want to have smaller size, etc. So it has to be discussed, and we can help definitely to develop that, but it's not commercialized per se right now.
Okay, thank you. So a global question for Marzia to go really from the lab to the clinical application. If you look back in the 5 last year, do you have any advice or share or to share with the team, or to do something differently? Because it's always a big step with all this research for clinical application. So it's more a global question. We could ask it to several people according to the roadmap that was drawn 5 years ago, but let's start with you.
Yes, thank you. Yes, obviously it's not easy because there are different languages from researchers and clinicians. We have the advantages that we are inside an hospital, so we started from the unmet clinical needs. So the first advice is to talk and stalk the clinicians, to take some information and to start from what they think that is an unmet clinical needs, and not from the technological point of view. The techniques and the methods could be only the methods to arrive to the clinical outcome. So you have to start from the needs and to also to think about the final clinical outcome. And that only in this way you could help really the clinicians and to work together. Every day is not really simple. In this time from COVID pandemic situation is not too easy, because also my lab that works in a hospital work only primarily only on COVID pandemic situation. But I hope that in some months this situation will be stopped. So my general advice is to start to talk about and to take to bring the jacket to the clinicians and to talk and to speak the same languages, because it's not so easy, but we have to not to have only the technological point of view.
Okay, thank you, Marzia.
Thank you.
So the next question is for Rudolf from AMIRES. So now we see this public-private partnership. Do you see that in the next European program, do you see an increase of proposition? And do you suggest to academic lab to already prospect and find SMEs to have established partnership to submit new grants?
Thanks a lot. Yes, indeed. We see, actually, it's not only between Horizon 2020 and Horizon Europe, this shift. We continuously see that Commission is pushed also by the Parliament for having as many of these beautiful and excellent inventions and innovations brought to the market. So indeed, this partnership is a let's say a key for, let's say, decision-making, whether to go for this type of public granting or not. What I was trying to also answer in this question is that we will not see only a public, that means research, private partnership, but we will see also a lot of private-public partnerships in terms of deployment of these technologies to the hospitals, to the healthcare systems. So that what we see clearly is more emphasized in this new program.
Okay, thank you. And for the last minute, I take last question also related to OrganTrans about the mechanical properties of the of the construct that will be how it will be accepted by the organ and the plasticity. So it's true that in this case the implants are very soft as they are hydrogel based, and they can be easily inserted by the host tissue. But in contrast, the mechanical properties will influence the cell differentiation, and this is really an important parameter during the maturation, but also at the final location to control all these mechanical cues. Okay, there are still some questions coming. With the Swiss time, it is 12 now, so we will close the third session. Maybe my colleague can put the slide, the last slide, so you know that this webinar series is part of the next generation organ-on-chip and organoid event that was postponed due to the sanitary situation, but it will physically take place on January 26-27 in Geneva at the campus Biotech. And in the meantime, you will have still 2 webinars, one on organ-on-chip for women health that will take place on September, and another one on November on emerging technology. This communication tool and platform for exchange will also maybe continue after the physical event, and we will really have this analysis after 1 year, but at least we had more than 200 registration for this event, so we really see this increased interest for regenerative medicine, and also, as it was mentioned several times, the transfer of existing technology for other fields that can be applied to RegMed. And now with already this panel of speakers, the OrganTrans network and all the attendees, it is really a nice basis to build up some new collaboration. So on my side, I really thank you for your participation, and I give the last word to Mariana.
Thank you, Jill, and thank you for all the speakers for their time in giving the presentations, and to all the attendees who could attend. You will just a final announcement. We will share with you the recording in the following days. Thank you all, and have a good day.